Okay, hi, I'm Jessica Faust. Thank you so much for being here today. I'm very thrilled to be at this point in my research and to be able to share it with all of you. So the title of my thesis, also before I start, there are treats in the back that Veronica provided, so if you need a snack, then feel free to find your way to the back room. So the title of my thesis is Bat Activity and Distribution in Coastal Southcentral Alaska Copper River Delta. So a little bit about what you will hear today. I'll begin by explaining a bit about bat distribution and diversity, some things you might not know about their distribution and diversity, and then some things that you probably do know about bat echolocation and nocturnality. We'll talk through bat habitat selection. I will propose to you why I think you should care about bats, and then funnel down to my study question and my predictions. Of course, we'll go through my methods, my analysis and results, and then kind of circle back to my questions and predictions in the discussion portion, and room for future studies. Of course, I have a lot of people to thank in my acknowledgments section, and then we will open it up for questions and answers. So, worldwide distribution of bats. Bats are more widespread and diverse than you may realize. They are found on every continent except for Antarctica. In orange there, that is the worldwide distribution of bats. And so there are over 1,200 species of bats, making up 20% of all mammal species. So that's a lot. I'm sure you've heard of fruit bats. This is the common short-nosed fruit bat. This is another fruit bat called the wrinkle-faced bat. And these are, I had to throw these in because they're just so hideous looking. All those contours on its face are used for echolocation. And they even have these really weird skin folds that they can pull all the way over their face. That even have like eye holes, like see-through eye holes. They are insane looking. Beyond fruit bats, I'm sure you know of this bat, but maybe not just from its face. Maybe now. This is a vampire bat. So it is feeding on the blood of a chicken's foot there. There are also carnivorous bat. This is a long-fingered bat, and it actually can dip down and catch fish and aquatic insects. So beyond carnivorous, blood-drinking, fruit-eating bats, actually 70% of bats are insectivorous. Bats use echolocation. So they use echolocation both for navigation, avoiding obstacles, but also for hunting. So if this bat is trying to catch this moth, it will send out a series of ultrasonic pulses. Those pulses will bounce off all the nearby objects, including the moth, and those will bounce back to the bat, and the bat can interpret it and then locate its prey. And they actually use a few different echolocation call patterns for different tasks. We'll get into that a little bit later. Why do they echolocate? Well, because most bats are nocturnal. So they need to travel and hunt in the darkness. So there are two prevailing hypotheses on why bats are nocturnal. The first we'll call the predation hypothesis. Bats are nocturnal to avoid predators who depend on the light to capture their prey. The second hypothesis is the competition hypothesis. So there is a chickadee eating an insect. So chickadees actually occupy a really similar ecological niche to insectivorous bats. So that means perhaps bats are nocturnal to avoid competition with other animals who occupy the same ecological niche. So if bats are nocturnal, what are they doing during the day? So similar to birds, they rest in roosts. So these are just spots where they rest. The roosts that they use during the day are called day roosts. And they use a variety of different types of structures for their day roosts. Worldwide, they have a lot of different structures. There are some bats in tropical regions that roost in like the folds of leaves. Bats use, of course, caves for day roosts. You may be familiar with a bridge in Austin that bats use as a day roost. So bridges, man-made structures, trees, cavities of large trees, also under bark of trees, inside rock crevices, things like that. Female bats and their pups, while they are nursing, create maternity colonies in maternity roosts. And since lactating bats and baby bats have less of an ability to thermoregulate, then these need to be a little bit warmer of like roost sites. So those are usually found in caves and man-made structures like houses or attics, things like that. Also, even though they are nocturnal, they use night roosts. So that's just a spot where they will rest in the middle of the night, and it's usually separate from their day roost. So a bat will wake up in their day roost, emerge, be active in the landscape for a while, then it will rest in its night roost, and then be active again before sunrise, return to the day roost before the sun rises. So we call that a bimodal activity pattern, because there are two bouts of foraging. So if I'm watching all night in a spot where bats are really active, after sunset I would expect to be seeing a lot more bats, a lot more bat activity. It would drop off toward the middle of the night when all the bats are using those night roosts and resting, and then a second activity pattern before sunrise. So that's bimodal, and in contrast, a unimodal activity pattern would be one main bout of foraging, and that will come up again a little bit later in the talk. But bimodal versus unimodal activity patterns. People often ask me what bats are doing in the winter. Bats can hibernate. They can actually enter like a daily hibernation even in summer, where they lower their metabolic rate, lower their body temperature, and can save energy during the day. But in the winter, they enter a much longer hibernation period, like you might think of when you hear the word hibernation. And they select these roosts. Once again, they need to be a bit more warmer and secure than their summer roosts, so they sometimes travel to their winter hibernacula, so they can travel short distances. So it's nothing like a long-term bird migration pattern that you would see, but they would just be, I guess the little brown bat can travel up to 200 kilometers to get to their winter hibernacula. Why do we care about bats? Well, I propose that bats are valuable to us. They are very effective at insect management. A bat can eat thousands of mosquitoes in one night, so that's good for us. Seed dispersal. Here we're talking about fruit bats. So of course, fruit bats ingest the fruit and deposit the seeds elsewhere, so that's good for the health of plant communities. There are certain bats that are pollinators. This is a lesser long-nosed bat, and it is pollinating the agave plant. So they are the only pollinator for agave, so we have bats to thank for tequila. They also pollinate cacao, mangoes, bananas, durian. So they're important to us because those might be things that we really like, but that's also important to our agriculture and our economy. Of course, up here we're talking about insectivorous bats. So insect management is the main reason that Alaskans might care about bats. But of course, there are always food web impacts. So if you affect one animal within the food web, this is a really kind of cartoon version of a food web of an insectivorous bat. But if anything happens to one animal in this system, then there are cascading effects on all the other animals. So bats are valuable. Bats are also threatened. They reproduce quite slowly. One bat can only really produce one pup in a year. So if anything happens to the population, then it's very hard to build those numbers back up. A recent study has found that bats are blind to buildings. So basically any really smooth vertical or horizontal surface is really difficult for bats to kind of interpret with echolocation. Horizontal smooth surfaces, bats will attempt to drink from because navigationally it seems almost like it's a water body. So then they can crash into smooth horizontal surfaces, break their jaws or get injured or worse. They also, if it's a smooth vertical surface, if it's something really shiny or glassy, then that can be really difficult to interpret with echolocation, so they crash into buildings. They're also attracted to wind turbines. So unfortunately, wind farms pose a threat to bats. They often crash into wind turbines, and so on wind farms you'll see plenty of dead bats. And there is something called white nose syndrome, which you might have heard of. This is a disease of hibernating bats, and it's caused by a fungus, Pseudogymnoascus destructans. You can see in the background, it builds up on the muzzle, giving it that white nose appearance, so hence the name. First detected in New York in 2006. It's since spread as far south as Mississippi and as far west-northwest as Bend, Washington. It arouses bats during hibernation, so it irritates them, causes them to wake up, and then they deplete all the energy that they have stored for hibernation, causing them to starve to death. It's killed over 5.5 million bats, and the little brown bat appears to be the most affected by this. It has completely wiped out populations of little brown bats, and it is not yet found in Alaska. So you just heard me mention little brown bats, and that's the main bat that I'll be talking about for the remainder of this talk. It's the main bat that we have in Alaska. We actually have 8 species of bats in Alaska, but in the other 7 are mostly in the, or have only been confirmed in really the southeast region. So little brown bats are the bats that we have confirmed that we have here in Southcentral Alaska. Here's a great photo by Veronica. So they're small. They're about the size of like a small rodent, a mouse or something, with wings that span out from the body about 8 to 10 inches. There's one in flight. So beyond bats being valuable and threatened, as we kind of already mentioned, this is an incredibly widespread species of bat. Here you have the countrywide range of the little brown bat, or I guess the continental range of the little brown bat. And animals with such a huge range often have really interesting things that can happen toward the edge of their range. So toward the edge of their range, animals can exhibit greater behavioral plasticity, so behaviors that you don't often see from this animal, or behavioral adaptations, broader tolerances to the environment than those in the center of the range. And since this is a nocturnal animal toward the northern edge of its range, we all know what it's like living in Alaska with daylight patterns. This presents a really interesting case study to see how bats deal with, for example, Alaska midnight sun toward the northern edge of its distribution. So this picture is of Cordova at midnight on solstice. So all my fieldwork was conducted in Cordova in the region called the Copper River Delta region. So my specific study questions are: what are the activity patterns of nocturnal Myotis lucifugus, or the little brown bat, in coastal South Central Alaska in the short nights in the weeks surrounding solstice? And what land cover characteristics are bats associated with in the northern coastal environment? I am not starting from scratch. Lots of work has been done on bats in northern latitudes, so I'll just walk through some findings of the literature. Of course, there's a lot more to it, but these are the findings that influenced my predictions. So in northern Scandinavia, at 65 degrees north, so about the same latitude as Fairbanks, and they were looking at northern bats in 24 hours of daylight. So they don't even have a short night there. They have continuous daylight through the night. Bats would wait until the darkest part of the night, and then they would forage in the shade of trees and mountains, but all of their activity was limited to about 2 hours in the middle of the summer. In Fairbanks, similarly, of course, Fairbanks also gets 24 hours of continuous daylight, and similarly, bats foraged for about 2 hours, but they foraged around above large willow trees. So not exactly in the shade of trees and mountains as in Scandinavia, but just above large willow trees. In the Northwest Territories, on short nights, bats maintained nocturnality. So this is the same latitude, essentially, that we have here. So there was a short night that was happening. And bats did not emerge before sunset or after sunrise. But they actually adopted a unimodal activity pattern. So if you remember what that means that they weren't taking, essentially, a night roost break in the middle of the night. They had a unimodal activity pattern. There's been some work done in the western portion of the Chugach National Forest on little brown bats. And in that study, bats were detected, so this is the Kenai area. In that study, bats were detected in every habitat type that they measured with no strong associations between bats and those habitats. So they discriminated these habitats by, they had sites that they called road sites, sites that were right along the road, sites that were right along water bodies. And there was no difference, essentially, statistically, on which bats preferred. They were found in high numbers in both road and water sites, as well as conifer sites, mixed and hardwood sites. But we do know a bit about general habitat selection, of course, in lower latitudes. So little brown bats often roost in trees, usually hardwood, but sometimes conifer. They've been found in conifer a couple of times, and rock crevices. Maternity roosts are found in caves and man-made structures. Of course, up here in the Southcentral region, there really aren't what you would call caves, so we are predicting that bats are mostly forming maternity roosts in man-made structures. Similarly, for winter hibernacula, caves and man-made structures, so we would predict perhaps man-made structures. And their foraging habitat. They were primarily foraging on the edges of cluttered shrublands near water. So here are the predictions that I went into my study with. I predicted that bats would maintain nocturnality and be active from sunset to sunrise. So essentially a unimodal activity pattern, but that did not go outside of the bounds of sunset and sunrise. I predicted to find feeding and drinking early in the activity period, and I predicted that bats will drink earlier than any other behavior. I predicted to find a higher association of bats in habitats with available water. Based on the pilot study in the western portion of the Chugach National Forest, I expected to find bats in all landscapes that I looked at, and I expected to find feeding and drinking in highly cluttered shrublands, such as Sitka alder, and I expected to find general echolocation in conifer forests, such as western hemlock areas and Sitka spruce areas, because those would provide canopy, potentially protecting them from predators, but also they're more easily navigable than like a cluttered shrub area. So they would use these as just travel corridors to get to their foraging sites. Okay, so I was working, as I said, in the Copper River Delta. If you look, this is the Alaskan range of the little brown bat, and the star right there is the Copper River Delta. Cordova, it's the region surrounding Cordova in the eastern portion of the Chugach National Forest. It's only accessible by plane or ferry, and once you're there, there's one road. But my project aims for to understand the distribution patterns and habitat association characteristics of little brown bats in the Copper River Delta, and to understand the behavioral patterns of these bats in this region. This is where it gets really cool. So how can we determine if bats are using an area? What you see attached to a branch here is actually a microphone. And this microphone, oh no! Well, there goes my picture. So this microphone is actually attached, maybe I can pull it up on here. There it is. Okay, so attached to the microphone is this device. This is called an SM3 song meter acoustic recording unit, and so it is attached to these microphones here, as you can see. It's about the size and weight of like a textbook, and it takes the same SD chips that you probably have in your digital camera, takes D-cell batteries, so these are incredibly easy to use and portable, and you can set it to turn itself on 1 hour before sunset, and to stay on until 1 hour after sunrise. And then essentially, if an ultrasonic sound occurs near the microphone, then the device will turn itself on and begin recording until the sound goes away. So I had 6 of these, and I set them at 17 sites across the region. So as I mentioned, there's only one road in Cordova, so I was kind of limited by road access. But I tried to distribute them in different areas throughout the landscape. And of course, the bridge is actually washed out here, so it's only about 31 miles of road access. And so these are the actual sites where I deployed these detectors. Each dot is a site. And you'll notice that they are color-coded. And that's because the first thing that I did when I deployed each detector was determine the land cover classification of the site. To do this, I used a dichotomous key, which comes from the existing vegetation classification mapping. Here it is. Existing vegetation classification mapping and inventory technical guide. So this is a common practice used by the Forest Service to determine the dominant vegetation in an area. And this gives you a land cover class based on the vegetation types that are dominant within a 10 meter radius. So if you can read this, I had one that was this blue color, this Mesic herbaceous site. I had 3 that were sweet gale, the yellow ones. 3 that were sitka alder, these pink ones. 1 that was sparse unvegetated, it's kind of hiding behind that green one. And 6 that were western hemlock sites, and 3 sitka spruce sites. So here's what they actually look like. This was the one Mesic herbaceous site. It is kind of a marshy wetlands dominated by sedges. This was the only site for sparse unvegetated, and it was really rocky right under the bridge, so within the 10 meter plot, the dominant vegetation type was no vegetation. Western hemlocks, as you might expect, is mostly hemlock trees that varied in size and height, usually somewhere between 6 and 9 inches of a diameter of breast height, but there were often a few that were bigger than that and a few that were smaller than that. And there are a few shrubs on the bottom here, so it's pretty clear between the trees. Sitka spruce is a similarly structured forest to the western hemlock sites, except for it was always associated with tall devil's club, so about a meter high of devil's club. Of course, sitka alder is dominated by taller shrubs, so this actually included both willow and alder shrubs, between 1 and 3 meters tall, very cluttered area. And then sweet gale also fell into a shrub dominance type, but as you can see, the shrubs are much shorter, it's a less cluttered area, and there's even conifer trees in the background there. So since I had 6 recorders, I was picking them up every 5 nights and moving them to different sites throughout the landscape. In addition to the land cover classes that I collected, I also noted whether water was present or absent within 25 meters of my microphone. And that 25 meters is actually the sensitivity of the microphone. So, and it changes based on how loud the sound is. But in general, a bat call can only be really detected within 25 meters of the microphone. Unless it is very quiet, then it needs to be closer in. So the acoustic files that I collected, if you remember this image, the actual acoustic files that I got back from the SM3 devices actually don't look so different from this cartoon here. Okay, so here is a file that I got back from an SM3. Okay, so you can see on the y-axis, this is frequency in kilohertz. And all of these ultrasonic blips, that is a bat passing through the area, so these are between 35 and 55,000 hertz. The time is here on the x-axis, and that is only 2 seconds. So as you can see, there's like 25 pulses in 2 seconds, and for the record, human hearing is here up to 20,000 hertz. So not only is this way outside of our hearing range, it also happens so fast that there is no way that we can interpret this without the help of these devices. And all this green fuzz here, just so you know, this is the noise that we probably would hear if we were walking through the woods. So wind and trees moving and water and all that kind of thing. That's what this is. So basically, similar to this, these blips are the bat emitting these ultrasonic pulses. And here's what it sounds like. So this is slowed down so we can actually hear it. Much lower in frequency now. That little half step was where it kind of messed up the rhythm. It'll get quiet. So that's really cool. As I mentioned before, bats have some characteristic calls that they associate with different tasks. So a bat that is hunting, that detects an insect, will emit what's called a feeding buzz. So basically what happens is it detects an insect and then it begins to emit pulses a bit more rapidly. And then the final part of the phase of the buzz is when they get much lower in pitch very rapidly and even more rapid than in this kind of region. Then we predict either it captures the insect or the insect escapes narrowly, and then it goes back to a regular kind of search phase call. This one is louder, so I don't want to hurt you guys. So check on the volume. I'll just lower this. This way. Then it goes back to the search phase call. In 2015, a study was published that actually identified the call pattern associated with drinking. Of course, it's no surprise that bats drink, right? But there had never been a call pattern that had been associated with this behavior. It examined 2 myotis species. This study was done in Europe. So it examined 2 myotis species, but not the little brown bat, unfortunately. So what I did is I read the description, and I looked at the description of how how kind of drinking buzzes work, and then I applied it to my research. But of course, it hasn't been paired with video footage. That was how they did the study, was they paired video footage of bats drinking to the ultrasonic sound that was happening simultaneously. But it hasn't been done with little brown bats. But here's how it works. Here's how it's described. So there's this search phase, as with everything else. Then in the approach, they start to come a little bit closer together. The drinking buzz, they're much closer together. And then the bat quits echolocating in here. And so that is as the bat is, so the bat emits this drinking buzz to assess the smoothness of the water, the distance, make sure it doesn't overshoot its target, then quits echolocating, and then there's a sound made here as it makes contact with water. And then I think of it like a stone that's being skipped. So it dips in, drinks some water, comes back up, emits another drinking buzz just to check where everything is, and then makes more contact with water. This is so much harder to hear, but I'm going to play it anyway. I'll turn it back up. This is quieter. You can hear some of those low frequency clickings towards the end. And then it goes back to the search phase. Okay, so as I said, there were 2 main things that I collected in each site. And those were the land cover class, and then the counts of these 3 behaviors. So each file that I got back from an SM3, I sorted into determining which behavior happened in each bat pass. So that means a bat pass and then since the land cover classes and the behaviors are all categorical, I used a residual analysis to find or to detect patterns between bat behaviors and land cover classes. So this is a table of the total counts of each of these behaviors, and then in parentheses here I have how many sites were in these vegetation classes. So for example, I had 1 site that fell into the music wet herbaceous class, and in all the nights that I surveyed it, there was only 1 general echolocation pass. I didn't detect drinking or feeding. So for the residual analysis, I couldn't use numbers that were too low, so I selected 5 or fewer vegetation classes I could not use. So that means these 2. So that meant that I only compared the shrub land cover types, the Sitka spruce, and the, I'm only changing the slide on my own computer. Okay. Oops. So that meant that I only compared shrub land cover types, Sitka alder and sweet gale, to our tree land cover types, which were western hemlock and Sitka spruce. For each of these individual cells, I calculated the adjusted standardized residual. I'm very happy to talk about that calculation maybe in the question and answer portion, but it takes a little while. But basically, we calculated the adjusted standardized residual, and then you can use it like a z-score in a z-table to find if each individual cell was significantly varying from the overall pattern. So let's explain what that means a little bit. So here's a table of this, it's a table of the same table that you just saw, but this is instead of the number of bat passes, each of these values is the adjusted standardized residual. So if we look just at general echolocation, GE, for Sitka spruce, the adjusted standardized residual value is 0.77. So then I take that and treat it like a z-value and see how significant that score is. So if you've ever used a z-table, I'll just walk you through. You go to the hundredths place, 0.07, go down to the tenths place. So this p-value is basically 0.22, which is not significant. So that is why Sitka spruce, this box here is not marked in red or blue, because that number was non-significant. So if it was a positive significant deviation, I use the color blue. If it was a negative significant deviation, I use the color red. And so how you can kind of read this in English is that in Sitka alder, look for the blue, bats are feeding more. In Sitka spruce, bats are drinking more. In sweetgale, bats are using general echolocation more. And in western hemlock, bats are drinking and feeding more. I also did an analysis for water availability. So I wanted to see if presence or absence of water had an effect on bat activity. So the y-axis here is the number of sites. The x-axis is the presence or absence of water. And the z-axis is number of bat passes on a logarithmic scale. So low is 0 to 10, medium is 10 to 100, high is 100 to 1,000. So a way to interpret this is there were 5 sites total that did not have water that had a very low number of bat passes. There were 4 sites total that had a high number of bat passes, between 100 and 1,000 bat passes, that had water. There were no sites without water that had a high number. So you can see that there is a trend, that there is more activity happening in sites with water than without water, but from a chi-square, the p-value was insignificant or not significant of 0.06. This is a figure of all the calls that I recorded during the sampling period. So how you can read this is this purple line is sunset, and this orange line is sunrise, and every single circle in here is a bat passing, is a bat being recorded. And so these are separated by individual night. Each line is a night. And then I color-coded them based on what type of habitat they were in. And the dots along here are where the detectors were. So you might look at this and you might say, oh, there's no pink happening in the beginning of June, between June 5th and June 10th. So I guess bats just don't use Sitka alder in the beginning of the month. But really, I didn't have a bat detector in a Sitka alder site at the beginning of the month, so that's important to know. And in general, you see, bats maintained nocturnality, right? There's no circles except for this one bat that was 1 minute early on June 11th, or whatever it was. But in general, all of the passes occurred between sunset and sunrise. And it actually looks pretty unimodal, right? There's, you don't see a gaping hole in the middle of here, in the middle of this, where you predict that bats are resting. But the more that I looked at this, and then the more that I looked at just the green, you look at just Sitka spruce, and actually western hemlock as well, the lighter green. It's hard to tell because there are so many passes in western hemlock sites, but it actually does drop off quite a bit here in the middle area. So if you look at that as a histogram, then this is bat calls binned by every 10 minutes after sunset, number of bat passes for the whole sampling period. And the white bars are western hemlock and Sitka spruce added together, and the black bars are all other vegetation classes. And what it looks like to me is that in western hemlock and Sitka spruce, this looks like a bimodal distribution or bimodal activity pattern. And for all other classes, it really doesn't show the same pattern. It's not quite a unimodal because it doesn't have, a peak toward the middle of the night or something like that, but it is kind of a steady activity pattern. Why could this be? Well, we already talked about our night roost explanation. Bats in other parts of the country use night roosts, so that could explain it. It would explain the drop in activity in the middle of the night, but it wouldn't really explain why bats are active at other sites at the same time. If bats are using night roosts, why do they only use night roosts in western hemlock and cypress spruce? I also think that there could be a day roost explanation. So if we think that maybe bats are using these conifer trees as their day roosts, then that would also have kind of a bimodal distribution pattern. If you put a human detector at the entrance to like the dorms, then you would also see a bimodal activity pattern as everybody leaves in the morning, goes somewhere else, and comes back toward the end of the day, right? So they would leave these areas earlier upon waking. They would pass through these areas to get to other sites for then perhaps their duration of nighttime activity, and then return to them late in the morning. There also could be explanations based on predators and prey. Maybe bats are only able to be active in areas with canopy cover in the beginning and end of the night, because that's when, sort of that twilight is brightest. So they use western hemlock and Sitka spruce in higher light levels, and then they can use other landscapes when it's a bit darker to hide from predators. Or maybe they're moving according to prey availability. Maybe prey is more available in those areas at dusk and dawn. Another interesting hypothesis came up in my last committee meeting, was presented by Jessica Reimer, perhaps, or not perhaps, but was presented by Jessica Reimer, which is that perhaps, so first of all, something really interesting is happening with female bats so something interesting is happening with female bats in this time of year, and that's that bats in mid-June and July are lactating or pregnant, right? So perhaps these black bars, what we're seeing, are female bats who are nursing, who are going back and forth to their roost throughout the night, and perhaps these white bars are male bats who have the luxury of taking a break in the middle of the night, don't have the same energetic constraints, and could perhaps be roosting in the conifer trees as well, whereas the female bats would be in a maternity colony. So this is a similar figure to the last one, but it doesn't have anything to do with the land cover class or the habitat type. This is actually all behaviors observed across all habitat type. So the white bars are the general echolocation. The orange bars are just feeding buzzes, and the blue bars are just drinking buzzes. And you can see that a lot of the feeding and drinking is happening right here in the beginning. There's also just a general peak in activity toward the beginning of the night. And toward the end of the night, I guess there's like an overall, almost a bimodal distribution, but it's still, the activity is still pretty high here in the middle. So it's not, they're not showing an overall bimodal activity pattern. But if we look just at drinking, here's the same figure of drinking as the last slide. It's just on smaller scale, so you can see more. So actually all of the drinking that's happening, almost, is within the first 90 minutes of the night. And I did a one-tail t-test to see if drinking activity was happening earlier than any other behavior, and it was a significant p-value. So basically bats are drinking, in sites where bats are drinking, bats drink earlier than any other behavior. So why could that be? Well, for male bats, it's a pretty easy explanation, right? There's a long day happening, they're in a roost, and they are losing water through evapotranspiration, meaning that just being in a warm, humid roost, they are losing water just by breathing, right? And so when they wake up, they're thirsty. For female bats, as I mentioned, the timing for parturition and lactation is late June, early July, so of course that places higher demands on water intake. So female bats could be prioritizing drinking upon waking because they're lactating. There's a similar pattern when it comes to bat feeding. I didn't know where else to put this picture, but I had to use it. So here is actually a histogram of bat feeding, just like the one that you saw a few slides ago, but on a larger scale so we can see what's happening. A lot of the bat feeding is happening within the first 2 hours. Most of the bat feeding is happening within, the first 60 to 90 minutes. But then there is feeding throughout the rest of the night.
You are the only participant in the conference.
Ooh, welcome. So let's circle back to my predictions and see how we did. First, I predicted that bats would maintain nocturnality and be active throughout the night from sunset to sunrise. Quick look at this, you see, yeah, that prediction was pretty much correct except for that one bat that was a little bit early. Bats maintained nocturnality for all of my sampling period. They weren't exactly active from sunset to sunrise. They actually quitted activity a little bit before sunrise, sometimes up to, I think the average end in activity was 67 minutes before sunrise. But they emerged very shortly after sunset. So we'll give that one a check mark. I predicted that feeding and drinking would be early in the activity pattern, in the activity period, and I predicted that bats will drink earlier than any other behavior. Yeah, a quick look at this, you can say that yes, that prediction was correct. I predicted a higher association of bats with habitats with available water. And if you remember this, this diagram, actually the p-value was less than significant. There is a trend, but it is not a statistically significant trend. So we'll leave that as a blank box. Not exact. We haven't exactly proven or disproved the null hypothesis there. Based on the pilot study, I expected to record bat presence in all landscapes. And actually, yes, in every single habitat type that I had, we had at least one bat passing through the area. And of course, there are some areas where there's much more activity. I predicted feeding and drinking would be in Sitka alder, and I predicted general echolocation would be in western hemlock and Sitka spruce. And actually, a more complicated pattern emerges than that, right? In Sitka alder, bats were feeding more, as we predicted, but in Sitka spruce, bats were drinking more. In sweetgale, actually, bats used general echolocation more, and in western hemlock, bats were feeding and drinking more. And that's where we got most of our calls were from western hemlock sites. So we'll leave those as kind of blank boxes, but diving further into this, if we just consider bats are feeding in Sitka alder and western hemlock, and they are just echolocating in sweetgale. It's interesting when we look at it from this map. It's just dots on a map. But it gets even more interesting when you look at it as a continuous landscape. So as I mentioned, I used these, I determined these land cover classes using the dichotomous key from the existing vegetation mapping project. And this area has actually been mapped using GIS technology. And I used the same colors that they did. So if you actually look at the entire landscape, the orange box is kind of the area that we've been looking at this entire time. But the entire area is classified in these colors. So if we remember, bats are feeding and drinking in pink and light green, and they're just generally echolocating in the peach yellowish color. Then it kind of starts to explain a little bit of a story, right? If they're feeding and drinking in the pink and light green, but all these areas are kind of near, just on the periphery of the sweetgale area, then you can think of the sweetgale area as either travel corridors to get to their feeding areas, or perhaps just peripheral areas where they pass through, they feed, and then they circle back and go back to their feeding area or something like that. Next steps that I see for this project. As I mentioned, drinking has not been confirmed for Myotis lucifugus for little brown bat, so I'd like to pair video footage with video footage of drinking bats with the ultrasonic sounds of drinking bats in this species, in order to put some confidence to the calls that I classified as drinking and to be able to apply it to future studies. I was only able to deploy recorders for 18, 19 nights, so I would be very interested in setting these out for longer in the region to determine how activities patterns change as the seasons change, right? So I set them out at a period that I found very interesting, right around solstice. But if we were able to leave them out for longer throughout the rest of the summer, as the time of sunset is changing more dramatically, then that would be very interesting. Paired water sites. So I had sites with water and I had sites without water, but they weren't identical in every way besides having water and not having water. So I would be interested in reproducing this work with paired sites that were more controlled for in every other aspect. Some work, if we could capture and attach radio transmitters to bats to be able to determine their roosts. That would be really interesting to be able to, first of all, locate a roost, and then locate or determine some habitat preferences for roosts. If we could follow them to a maternity colony, if we could follow them to a winter hibernacula, all of these things would be very interesting to determine what they're doing in the region. Insect trapping at each site. So of course we know a bit about insect activity and insect habitat, but to be able to trap at each site to determine insect availability, prey availability, then that might help fill in some of the blanks of what they're doing in the landscape as well. I have a lot of people to thank. Thank you so much to my committee members. Thank you, Doug, Matt, Jessica Reimer, Jessica Isles, joining us from Puerto Rico. Thank you to everyone from the Forest Service, especially in the Cordova Ranger District, Melissa and Aaron Cooper. Thank you, Susan Loeb. Thank you to the Department of Fish and Game for lending us some equipment. Thanks to all my field, to everyone who helped me in the field, Janine Mistrick, Sarah Cooperman, Keegan Crowley, Sammy Buchner. Thank you to the whole CAUSE Lab. Thank you to my friends and family who are here today. I can now open the floor for questions.
Thank you. Any questions?
Yeah.
Yeah, I have two questions.
Sure.
Can you fill me in a little bit on the background of why you chose your study site? Why that specific area?
So that specific area, I think it has an interesting landscape to it. You know, it's coastally dominated, which bats are not exactly more associated with usually, but there have been a lot of first-hand accounts of bats being in the region. So, so that made Cordova a really interesting place to work. It was also a fun place to visit and do field work for the summer. But it's really there aren't a lot of man-made structures if you've ever been to Cordova. It's pretty undeveloped, so besides the main part of the city. So when I work in Anchorage or when I deploy detectors in Anchorage, then you can assume that most of the roosts that they're using are in man-made structures, whereas in Cordova I was hoping to perhaps find bats that are roosting in more natural structures.
And secondly, what's your most exciting novel finding that differentiates your science from the previously published literature? What do you think? What's the best part of your thesis?
I think the most interesting part for me is being able to apply the drinking characterization to my work. That really hasn't been it hasn't been used in this species at all yet, and it's brand new. So I think that's what I'm most excited about.
Thank you.
Yeah.
Yeah, the acoustic recordings are really interesting. So that's just Myotis specificus, right? And did you detect other bats with those acoustic recordings, or ones that you couldn't identify that you figured those are not Myotis?
So it's really interesting, especially in Alaska, because if you use, so there are softwares. The software that I used, that I showed all those calls from, is the Kaleidoscope software. And it has a function where it can sort your calls based on species. And in Alaska, it has no idea what to do with these calls. It calls them like Indiana bats and all kinds of other things because they seem to have a little bit of a different dialect up here. So they're echolocating at slightly different frequencies than is often associated with the species. So from this, I couldn't determine if it was one species or several, but based on what I know about, or based on what we think we know about, species in the region, we assumed that all were little brown bats that we detected. but none were vastly different, I guess, in the echolocation pattern. Yeah.
Can you differentiate individuals with the software? Or is it one bat that's flying around for 25 meters within that circumference
Great
for 2
question.
hours?
Isn't that annoying? No, you can't tell if it's 25 bats flying by or if it's a bat flying by 25 times. But what I can tell you is that bats are active more in that area, right? It might be one bat that's doing all the activity, but they're using it more.
Yeah, more
Yeah.
questions. So you mentioned you want to measure the insect population because you can see how that would influence the feeding.
Mm-hmm.
Are there known patterns and like bursts of insect numbers during that time of year or in that area? Is anyone just concurrent studies? Or I just curious, like, are there more mosquitoes one night, or was there a temperature difference that day, or wetter or raining? You know, that you could use to correlate your data.
Yeah, so I actually did study, or I did, some simple linear regressions on. I made some additional slides just in case you'd ask me that. So this is the ambient air temperature. The red is the maximum temperature, blue is the minimum temperature, and then the black dotted line is the.
Jessica.
It's not me. Oh, thank you. Now you see what I'm talking about. So you can see the ambient air temperature, and so I did linear regressions comparing bat activity, the mean temperature, the minimum temperature, the centimeters rainfall, and for all of those, we did not detect a difference in bat activity. And that actually rainfall and temperature does significantly affect both bat activity and, of course, insect availability. But since it didn't vary dramatically in these 18 days that I surveyed, then we actually predicted that we wouldn't find a significant difference in bat activity.
Okay, if there's no more, yes?
Yeah.
So you talked about the maternity roosts being generally in caves or man-made structures. So where do you suppose that they're, without a lot of man-made structures in Cordova, are there cave areas, or like where do you think they might be roosting in that scenario?
Yeah, so I think it is quite possible that they are all either near town or in the Forest Service cabins that are out there, and some of the structures that are found out in the landscape. So that would be my best guess, but it's hard to say. Yeah.
So following up on that, has anyone done captures out there, or is it possible that it's all males using tree roots and there are no reproductive females out there? There's no maternity colony sites?
Yeah, so I haven't found any identification of sort of what the ratio of sexes would be out there. And so that would be really interesting to find, especially in the sites that I use for future studies, to do captures and find out, how it shakes out with the gender ratios. Yeah.
Can you, once I saw that slide pop up about residual analysis, I was just wondering if you could explain a little bit more about what that is and why you use that approach versus like a generalized linear model for like count data or proportional data.
Yeah, so since these are categorical and they're not quantitative, that is why I used the residual analysis. So these are the equations. Basically, I have a whole math lesson I can walk through on how this works. But basically, you want to find out if each individual cell varies significantly from the overall pattern. And so you start with just the standardized residual, and you divide it by the square root of the variance. So finding all that. And it's because these are categorical, and it's also because there are low sample sizes that make these the
You
most
are the only participant in the conference.
Yeah.
So following up on Paul's, so that tells you if they're different from each other, but it won't take into account seasonal variation. So when you look at what you showed us before that table, all of the dates that were really late in the season had a lot of activity. So if you only look at your hemlock sites late in the season, they'll be significantly different, but that might not be because they're hemlock, it's because they were surveyed later in the season. And so is there any way with the residual analysis to account for that, I guess?
I guess the way that I saw it with the western hemlock being more more active in late in the season is based on where I put the detector. So activity might change in those 2.5, 3 weeks. But that particular site that I set it at for those last 5 days was called Nirvana Park, and it had more bat passes than any other site. It had over 1,000 bat passes in that area, and so that, so it's, if you look at, I guess, where's that slide? Too many animations. So if you look at this and you see, oh, it looks like bats are really using this week, and they're much less active in these weeks. I think it actually had more to do with where I placed the detector in that particular week than necessarily what bats are doing.
So again, does that residual analysis let you account for things like site or date or anything like that to account for some of those things that might be also playing into the difference in activity?
So based on my understanding, it wouldn't control for those things. Yeah.
If you had to do it all again, would you change your experimental design at all, and how would you do it so you'd be more confident with the results?
Yeah, so I mentioned the sort of the water site, so I think it would be really interesting to change the design a little bit to set a detector that's like within, say, 25 meters or 10 meters of a water body, and then set another detector, let's say 50 meters just straight back from that, in order to, and try and select sites that are incredibly similar in vegetation type and clutter and in all other factors, so that then I can be more confident in how bats are, or whether or not bats are used, or how important water is to bats. In addition to that, I think, of course, there's issues with how many in each category of land cover that I have. There's Music Weather Herbaceous, where I only have 1 site. There's Western Hemlock, where I have 6 sites. So, of course, I would like to perhaps use maybe a more randomized site selection pattern, but use, discriminate per site so that I have kind of an equal number of sites in each category to be able to use that. And then I also think that, I can collect every characteristic of a habitat. I can measure each tree, each tree's DBH and height, and all kinds of things like that, but when you put it all together, then it won't necessarily tell you how much bats are using the habitat. For example, I don't think that specific vegetation species is really that important to bats, but I think that probably the structure is much more important to bats. Because I don't think they're out there with the dichotomous key deciding where to go based on specific species, but that those specific species might be associated with insects and things like that, so it might change. So that would be, yeah, I think that if I were to go back again, then I could, I would like to replicate the number of sites in each category, as well as controlling for water more. Yeah.
There's quite a few private and recreational structures in that corridor.
Mm-hmm.
Did you investigate any correlation between your site distance from structures and bat activity, or do you have any sense where all of your sites kind of similarly distant from structures?
So if we look at kind of the map of where these were at, yeah. You can see some are much closer to town, right? So on this side, these are much closer to the actual, the city structures. And then out here, as you mentioned, there are cabins, every few, I don't know how to put it. They're kind of dispersed along the highway, and some are further back. Some are a 6-mile hike to get to, and some are a 1-mile hike to get to. So I didn't do any kind of analysis measuring distance of site to, for example, Forest Service cabins. But that's kind of my general interpretation of how close these sites were to man-made structures.
Any other questions? Okay, thank you.